Great. Well, thank you very much. So my name is Daryl Gohl. I lead a group at the University of Minnesota Genomics Center called the UMGC Innovation Lab. And I'm going to tell you today about our work on developing RNA interference-based genetic biocontrol tools for zebra mussels. So before I get started, I just wanted to introduce the team that's been doing this work. We've been working with Dr. Mike McCartney, Dr. Scott Ballantyne from University of Wisconsin River Falls, Lindsay O'Brien, who works in my research group here at the UMGC, and Dr. Victor Hernandez Lizarraga, who is a new postdoctoral researcher with us. So while there's been a real explosion of interest in genetic biocontrol methods that's really been driven by new gene editing tools like CRISPR. These genetic methods to control undesired populations have actually been around and been employed for many decades now. So one example of this is the eradication of screwworms from North America. So this was actually work that was started back in the 1950s, where scientists developed a method to irradiate and sterilize male flies. And then these flies would be distributed throughout the population, where they would compete with wild males to mate with females. And then any females that had mated with these irradiated flies would give rise to viable offspring. And using this method, as you can see in this graphic, screwworms were actually pushed out of North America down into Mexico, and ultimately all the way down to the Isthmus of Panama, where still today this genetic barrier is being actively maintained. So every week, millions of these sterilized flies are dropped from airplanes along this border to prevent the spread of screwworms northward. So more recently, new genome editing tools like CRISPR have enabled more targeted and novel methods for developing genetic biocontrol tools in a variety of different organisms. One such approach is this engineered genetic incompatibility approach that's been developed by Mike Smansky's group here at the University of Minnesota. Another commonly used another technique that's been the subject of a lot of research are gene drive approaches. So these have been proof of concept gene drives have been shown to work in insects such as mosquitoes. The way that gene drives work is they usually contain an element that modifies a trait of the animal. So commonly with insect species, you would create a genetic change that causes a strain to only produce males. And then gene drives have an additional element that enables this tool to basically spread very quickly throughout the population. And so you can imagine if a population, you have this strain that only can produce males, and that spreads very quickly throughout the population, that can lead to a population crash because there are no more females to propagate the population. So these tools have shown a lot of promise in lab-based studies, but they haven't yet been applied in the wild. So we're actually taking a somewhat different approach with zebra mussels. So if you saw Ben Minerick's talk earlier today, he talked about the methods that he's been using or that he's been trying to develop to cultivate mussels in the lab. And so it's sort of a great irony that these mussels spread so much throughout the lakes and rivers in the wild, but we can't actually grow them and cultivate them for multiple generations in a lab setting at this point. So Ben gave a great talk earlier about how he's doing work to optimize the mussel's diet and to try to get to the point where we could stably maintain mussels over many generations. And if we can do that, then we could potentially start to think about employing some of these approaches like gene drives that rely on making heritable changes to the mussel's genome. But in the meantime, we're sort of stuck with this question of how do we genetically manipulate an organism that we can't robustly cultivate in the lab? And so we've turned to an approach called RNA interference. RNA interference was initially discovered in this nematode worm called C. elegans, and it was found that you could feed this worm either double-stranded RNA. You could inject double-stranded RNA in the worm, or you could feed it bacteria, its normal food source, that express double-stranded RNA. And this exposure to the double-stranded RNA molecules would cause silencing of genes within the worm. So on the bottom here, there's a little graphic that shows a worm tissue that's expressing a green fluorescent protein, and then on the right, when that worm is fed bacteria that express a double-stranded RNA targeting that green fluorescent protein, the most of that green expression goes away. And so this was a very exciting discovery, a new way of manipulating gene expression in a wide variety of organisms. And it's subsequently, so it's thought that this system, this gene silencing system, actually arose as sort of an ancient innate immune response, or an ancient antiviral defense mechanism. So double-stranded RNA is not typically found within cells, but it can be a hallmark of viral genomes. So there are double-stranded RNA viruses. And so when double-stranded RNA is detected in a cell, there's this whole machinery of proteins that basically detects the double-stranded RNA. So there's a molecule called dicer that binds to and detects this double-stranded RNA, chops it up into small pieces, and then those pieces are loaded into another complex called RIS that basically searches the cell for any other transcripts, any other RNA molecules that have homology to that double-stranded RNA, and then it will either chop them up or translationally silence them. So in effect, what we're doing by trying to use RNAi in zebra mussels is using this machinery, this sort of innate immune response, to trick the mussels into mounting a response against and silencing their own critical genes. So RNA interference has been shown to work in a wide variety of different organisms. So I have some examples here from fruit flies, where you can see that expressing RNAi against this gene called eyes absent gives a very similar phenotype to a mutation in that gene, causes the fly to have no eye. RNAi has also been shown to work in other bivalves, such as oysters, which gives us increased confidence that we could get this system to work in zebra mussels. And even humans also have RNAi pathways. So the FDA has now approved something like 5 therapies that are based on RNA interference as a mechanism of action to treat human diseases. Since its discovery, RNA interference has also been used to develop tools to control pest species or disease vectors. So there's been RNAi-based mosquito larvicide that's been developed, and that where mosquitoes are fed algae that express double-stranded RNA against target mosquito genes, and this can be used to kill the larvae so that they don't develop into adults. There's also a plant pesticide that is now based on an RNA interference mechanism. So there's an EPA registered crop that expresses an RNAi molecule against the corn rootworm, and this is used to control an agricultural pest. So we're working on taking this approach in zebra mussels, where we introduce zebra mussel target genes into microbial cells, and then feed these microbial cells that express double-stranded RNA against zebra mussel targets, and then score either effects on zebra mussel physiology, or effects, or try to quantify the effects on gene expression in zebra mussels, with the goal of ultimately developing tools that can decrease the fitness of zebra mussels, and could potentially be used for genetic biocontrol. So some of the advantages of this approach, first, RNAi provides a means to manipulate the biology of this organism that we currently can't cultivate in the lab. Second, it has the potential to be highly species specific. So because the specificity of an RNA interference construct depends on base pairing between the interfering RNAs and the RNA that's expressed by the organism, we can really dial these in and get very specifically target zebra mussels and hopefully minimize or eliminate off-target effects in other species. So that has some potential advantages relative to chemical or physical controls that might have broader impacts. RNAi also has flexible options for deployment. So if you saw the talk earlier from Jim Wambush and Chris Merkis, they described basically expressing this, working to express these RNAi constructs in algae, and then put them into bait that they could feed to fish to control their populations. So you can express and deliver it. So that would be expressing and delivering in an inactivated microbe. But one could also potentially deliver it as a chemical agent, as the double-stranded RNA itself, or potentially in replicating microbes, which might persist for longer in the environment, which would have, the pro of potentially being a more potent and long-lasting control agent, but have the con of having a genetically modified organism that can replicate and be released in the environment. And then also, because this gives us a tool to manipulate zebra mussel in a targeted manner, one could also imagine pre-treating mussels with these RNAi constructs and then releasing them, sort of similar to what's done with the sterile insect technique. And then finally, this wouldn't actually change the genetic code of the zebra mussels. So when we started this project, there were a very large number of questions, and I'm not going to read through this long list, but a large number of questions that we need to answer to determine if this approach is going to be viable as a biocontrol tool. And I've sort of just broadly categorized these into proof of concept. So does this system exist and function in zebra mussels, and can we demonstrate that we can use an RNA interference-based mechanism to affect gene expression in zebra mussels? And then the next phase of the project is optimization. So figuring out which targets, which microbes, which mechanisms of delivery are able to best induce phenotypes in zebra mussels, and also starting to answer questions like, are there off-target effects in other related mussels or other cohabitating species? And then the final phase of the project will be sort of permitting, field testing, and ultimately deploying these reagents. And there are a number of both technical questions. So can we produce these reagents at a scale that would enable treatment of large water bodies? But there are also regulatory and social questions. So there aren't really regulatory pathways that have been defined for tools such as these. So this will, for any of these genetic biocontrol tools, there'll be a process of working with regulatory agencies to define those processes, and also involving key stakeholders and the general public in making decisions of how to responsibly and safely use these tools. So the project that we're doing grows out of work that we did to sequence the zebra mussel genome. This was published in recent years, and the zebra mussel genome project was a really large group effort. It involved Mazer scientists. So this project was initiated and led by Dr. Mike McCartney, and then researchers from my group in the University of Minnesota Genomics Center, and a number of other labs at the University of Minnesota, and lab and industry partners outside the university. We used a wide variety of DNA sequencing technologies to sequence an individual zebra mussel that came from the Duluth Harbor And we ultimately generated a chromosome scale genome, super high quality genome assembly for zebra mussels. And so this genome was able to help us start to answer some of these questions. So for the first question, do zebra mussels actually have the machinery to carry out RNA interference? And then the second question, what genes and sequences would make good potential biocontrol targets? So this is a graphic that was prepared by Scott Ballantyne and one of his classes at University of Wisconsin River Falls, where they searched the zebra mussel genome for the components of the RNA interference pathway. And they found homologs of all of these different components that are known to be essential for RNA interference in other organisms. So the answer to that first question seems to be yes, that zebra mussels do in fact have the machinery, at least encoded in their genome, to carry out RNA interference. And then in the context of the Genome Project, we also did a number of experiments that were focused on trying to understand processes that we thought would be important for the survival of zebra mussels or their dispersion. So we generated a list of genes that are required, that are expressed in the mantle, which is the tissue that makes the shell of zebra mussels. So we have basically a catalog of all the genes that form the shell. We also were able to generate a similar catalog of genes that make up the byssal threads, or the attachment fibers that they use to attach to surfaces. And so based on some of these experiments and these target genes, and then a variety of other analyses that we've done of the zebra mussel genome, we've identified and prioritized a list of about 150 target genes whose disruption we think could potentially impair zebra mussel fitness. And that's genes that are in these categories of shell formation, attachment fibers, but also things involved in really basic cell biological processes, sort of these essential genes that we think will essentially be lethal if we knock them down. And also genes that are involved in processes like reproduction. So, the next question is, okay, they seem to have the machinery, but is it actually functional? And so we've tried to attack this problem in a couple of different ways. So in past years, I've talked a little bit about some feeding experiments where we've been expressing double-stranded RNA in microbes and feeding it to zebra mussels, looking for knockdown. But we actually realized that there's a more direct way to do this experiment, and that's by just basically injecting double-stranded RNA directly into zebra mussel tissues. And so this is work that's been done by Victor and Lindsay. So they developed a method where they can basically take mussels and put them in a little dish, like what's shown here. And then what will happen is the mussels will open up to begin filtering. So they open up their shell and stick out their siphon. And then what they do is jam a plastic pipette tip, which is this little thing that's being jammed into that gap, to basically shim the shell open so that the mussel can't close it. And then that gives them an opening to be able to insert a needle and inject double-stranded RNA directly into the tissue of the mussel. And what we're targeting are actually the muscles. So these two muscle tissues that are right at the kind of accessible right near the shell opening, the posterior adductor and posterior abyssal retractor muscles that we're targeting for injection. And so what this looks like is shown here. So what you see on this slide these are mussels that have been dissected out of their shells. And so you can see the byssal thread attachment fibers hanging off of the right side of the mussel. You can see these sort of whitish blobs on the left side, and those are those two mussels that I had highlighted on the previous slide. And so these experiments, or the images shown on the right are imaging a fluorescent dye that we're injecting into that tissue, which is called calcine. So the green glowing spots that you can see are calcine injected into those muscle tissues. And that just gives us a way of tracking and making sure that we've successfully, hit the target, we've successfully injected into the tissue. And then we've been doing experiments where we're injecting either, doing a sham injection or no injection, or injecting a double-stranded RNA that's targeting a zebra mussel gene. And so in this example, we've injected a double-stranded RNA that targets a gene called RPABC5, which is a component of the RNA polymerase, which is an essential gene that's required for basically expression of all other genes. And so what we do is Victor has injected this construct, and then we wait 24 hours, and then we harvest the tissues, extract the RNA, and then do a technique called quantitative RT-PCR or qPCR in order to quantify the levels of this transcript. And so what you can see is that in the injected mussels, the mussels that have been injected with this double-stranded RNA, there's about a 4-fold reduction of transcript level, which suggests that the double-stranded RNA is indeed knocking down the gene in this tissue. So now that we have some evidence that there's a functional RNAi system in zebra mussels, the next step is to try to engineer microbial strains that express different RNAi triggers to then see if we can feed the mussels these microbes and detect effects on the actual survival or health of the mussel. So we're taking two different approaches to engineering strains that express RNAi constructs. In one case, we're engineering bacterial strains, so that's shown on the top here. So we have a strain of E. coli where we insert a target gene into a plasmid, put it in the E. coli, and then we can induce expression of that target gene. But we're also trying to engineer an algal species, so Chlamydomonas reinhardtii, where we're inserting a target gene into the chloroplast genome of the algae, and then trying to use double-stranded RNA expressed from that algal strain to knock down gene expression in zebra mussels. And we actually think because algae is a preferred food source for zebra mussels, that approach might be more successful than using bacteria. So we've shown that we've been able to construct a number of these strains. So Lindsay and Margaret and Scott and also some of Scott's students have over the past few years constructed about 30 or so E. coli expression strains that express double-stranded RNA. We've shown that we can induce the double-stranded RNA production in these strains. And these strains target a wide variety of processes. So they target a number of what we think are likely essential genes that if we're successful in knocking them down, could essentially kill the mussel. We've also got RNAi constructs that target the byssal thread attachment fibers, as well as targets or constructs that target serotonin signaling, which is important in reproduction, spawning in zebra mussels. And so we've also been working to make expression strains in algae, and this is technically more difficult. So it's relatively easy for us to make these bacterial strains, but it's taken quite a bit of work and quite a bit of troubleshooting to figure out how to engineer algae. But recently we've started to get some of our first RNAi strains for algae. And so what's shown here on the left is a graphic of basically the process that we're using to make these strains. So there's a really clever genetic engineering method that other groups have developed that has some selection strategies where essentially you knock out a gene that is required for photosynthesis in the chloroplast, and then you can basically select for your gene to be recombined into the chloroplast genome by requiring the strains to grow under light. So it's only when you replace and correct this defect in photosynthesis that you're able to that the algae is able to grow. And so we've gotten our first few target genes into algae, and we've also been able to show that those can be expressed. So the other major thing that we've been working on over the past couple of years is developing ways to measure the effects of RNAi. So, once we start to generate these constructs that we can feed to zebra mussels, how do we measure their effects, and try to ascertain, how they're impacting the mussel? So we've been working on a variety of different assays, developing methods for measuring mussel growth over time, developing methods to measure their attachment to surfaces or their ability to filter bacteria or algae. So basically, how effectively are they feeding? We've also been working on methods to quantify either gonad development or spawning, so we could begin to look at some of those constructs that target reproduction in zebra mussels. So I'll tell you about one of these assays. I've talked about this in the past, but this is a test that we sort of jokingly call zebra mussel Yahtzee. So this is a method that we use to measure attachment in zebra mussels. And the way that it works is really simple. We basically detach the zebra mussels from their substrate, put them into glass jars, incubate for a period of time, and then we take and invert the jar and we count how many mussels have attached. And what we found is that if you do this sort of sequentially, we can basically do this every day, come in, detach the mussels from the surface, let them sit overnight, next morning play Yahtzee, see how many have attached. And the mussels will really reliably reattach over the course of many days. And so this is an assay that we can sort of do sequentially over time to quantify the attachment to surfaces. And we're envisioning this as both a test that could tell us, how well those byssal thread attachment fibers are working. But in addition, it also gives us information about the overall health of the zebra mussels. So we've done this experiment where we've taken mussels and we've exposed them to different doses of copper sulfate, which is a known toxin. If you saw Diane Waller's talk earlier, she talked about this chemical compound that's used to control mussel species. And then we assayed them and asked, how well are they reattaching? And what we found is that there was basically a dose-dependent effect. So as we added more copper sulfate, the mussels did worse and worse at reattaching to the surface. So not only is this an assay for attachment directly, but it's also could be used as a proxy for the health and well-being of the zebra mussels. So we've done a bunch of work with the bacterial constructs that we've made. And this is work from Lindsay O'Brien, where we've basically done this repetitive reattachment assay. And the way that this works is we detach the mussels, we let them sit for a period of time, and then we assay attachment. We do that a couple times to establish a baseline. So you can see here is, this is a pretty robust phenotype. Like these mussels attach, 80 to 90% of the mussels will reattach. And then what we do is we have a dosing period where we dose with one of our RNAi constructs. So we add the microbes, and the zebra mussels will actually clear them. So when we add them, the solution is cloudy. Come back in the morning, they've cleared the bacteria out of the water. So we do that for several days while continuing to measure their reattachment, and then we have sort of a washout period where we look post dosing. Do they recover or is there a decline? And so the data shown here is for either mussels that have not been dosed, so in the dark green line, or mussels that have been dosed with a construct targeting GFP, that green fluorescent protein, which is a gene that zebra mussels don't have. So effectively, this is a non-targeting control. It's just saying, if we add the bacteria, do we see an effect? And then what we can do is start to dose with constructs targeting zebra mussel genes. So that's, there's a particular construct shown here in the blue line. And what we see is that in general, for most of these constructs, we don't see a very large effect. So this is quantified here across a large number of constructs. So the controls are shown on the left, and then these are feeding with different constructs, different bacterial constructs that express RNAi genes. So in our initial round of experiments, at least, we haven't seen a big attachment defect in feeding microbes expressing these RNAi constructs. What we're doing now is gearing up to do this with Chlamydomonas, which, as I mentioned, is a better food source for zebra mussels. And then there are also a number of other features of these constructs that could affect how well they're able to be processed by the zebra mussel RNAi machinery. So ongoing work is basically trying to understand what features of the double-stranded RNA are necessary to induce a robust phenotype, and then what are the best delivery mechanisms to actually, other than injection, to get it into the tissues and to see an effect. In the next phase of this project, we were just recently awarded a grant from the Department of Defense. So they have this program called the Strategic Environmental Research and Development Program. DoD is a really large land manager, and they have impacts on both their infrastructure and the ecology of the lands that they're tasked with managing from invasive species. And so we're really lucky to get funding from them, and this will fund our work over the next 4 years to continue working on developing these RNAi-based genetic biocontrol methods. In this project, we're collaborating with scientists from the USGS down in Missouri, who have expertise on doing work in cell culture, which we hope will help accelerate our screening of different target genes. And they also have the ability to grow a variety of different native mussel species. So eventually, we hope to collaborate with them to try to assess potential off-target effects as well. And so this grant is really going to focus on the next stage of this project, which is, what are the best triggers? What are the best delivery mechanisms? Are there off-target effects? And hopefully will kind of move us along toward developing something that could be potentially a field deployable genetic biocontrol tool. So with that, I'd just like to thank all of my colleagues at the University of Minnesota Genomics Center, especially my colleagues in the Innovation Lab, and all of our collaborators who have worked with us on this project, and our funders at NASERC and now the Department of Defense. And I'm happy to take any questions.
I was wondering, so thinking about, the algae culture and how you're going to be feeding them, do zebra mussels, like native mussels, produce a pseudofeces, and how would that influence, how do we confirm that they're consuming this?
They do, yeah. And that's actually one of the challenges, or one of the potential, reasons why these feeding experiments that we've been doing may not be working, or haven't been working, right? So it's known that zebra mussels can selectively filter food particles and particles of different sizes, and that they do basically deposit. So they'll filter and digest a subset of what's kind of running over their gills, but then they'll also basically shunt and deposit in pseudofeces these other undesired particles. So we don't actually know how the bacteria that we're feeding them are partitioning, if they are being partitioned into pseudofeces. There are some assays that we could potentially use to try to figure that out. Actually, in one of the earlier talks on developing baits and RNAi tools in carp, they use a dye basically to tell if the carp have been ingesting these baits. I think something like that might be a way of monitoring whether it's being partitioned in the pseudofeces or whether it's actually being digested. But yeah, we're hopeful that algae are much less likely to be much more likely to be consumed and less likely to be sort of filtered out.
When you get to the final phase, is there a way to test non-targets genetically to evaluate the effects of the RNAi?
there are a few different things that can be done at the design phase, the current phase that we're at. Once we start to zero in on promising targets, there's a lot of sequence information out there. So one simple thing you can start to do is basically just query your sequence against all the other published things that are out there that we know to exist in aquatic ecosystems. And so you can sort of rule certain sequences out based on the fact that they might exist in other organisms. It's also true though that not every species has, not even close to every species has been sequenced, right? And so at some stage, that'll need to be really empirically tested. The approach that I think will most likely be taken will be to, sort of make lab-based mesocosm systems. So these are somewhat simplified systems, but systems that are designed to have some diversity that mimics the environment, because there are a number of questions, right? So there's one, is the RNAi tool potentially affecting off-target species, right? And causing, changes in gene expression or causing certain organisms to be negatively affected. But there's also the potential for, if we're thinking about delivering this as an algae, there's also a question, does that actually survive? And does it survive in the context of all these other organisms that might be consuming it or affecting what you put in, right? I think those sorts of studies, in a controlled lab setting where you try to mimic, either test certain targeted species or try to mimic a much more complex ecosystem, and then look for effects on population structure or off-target species, are going to be necessary before moving into a broader deployment.
So thinking about your research, what do you think is the most promising targets for genetic biocontrol in zebra mussels?
Yeah, it's a good question. I mean, in one sense, some of these genes that target processes that are really specific to zebra mussels, like, the byssal thread formation, right? There are a limited number of animals that, make these attachment fibers. Those could be good, if you want to avoid off targets. I actually think one of the challenges we have right now is that, it's relatively difficult when you have an adult animal, you're trying to manipulate the expression of genes. There's a lot of redundancy in biological systems, and a lot of, it can be relatively difficult to actually make an intervention that will kill the adult animal. Even these things like, the RNA polymerase gene that I mentioned, which is, a core component of keeping cells functioning, we would probably have to keep it turned off for a large period of time right before we were able to actually kill the mussel. One of the things I think might be promising though is, applying these tools when the mussels are developing, right? So earlier in Ben Minerich's talk, when he was talking about, how they have these kind of fragile stages early in larval development where it's very difficult to keep them alive, at least in the lab. The impacts of affecting gene expression during those critical developmental windows can actually be really huge. And so, I think feeding these, when there are lots of larvae in the lake, for example, and knocking down a critical gene during a critical window in development, that could really be an approach that could have a major impact. Now, what we need is for the work that Ben is doing to mature, where we can get lots of larvae in the lab that we can start to run these tests during those stages of development.
Are there any similar ongoing efforts to develop genetic biocontrol tools for other types of invasive species found in Minnesota?
Well, so we've actually been working with a group of other researchers, both across the country here in the U.S. and even internationally, who are working on developing genetic biocontrol tools for different invasive freshwater mussel species. So we've been working with a group that's been working on quagga mussels, and also a group that down in Brazil that's working on the invasive golden mussels. So this is like a mussel that originated in Asia that's really infested the Amazon River basin and is causing all sorts of ecological impacts there. And actually, each of these groups is taking slightly different approaches. So the golden mussels, there are struggles with growing all these different species in the lab. The golden mussels have the advantage that they actually spawn year-round, like they live in a more temperate area, and so it's a little bit easier to kind of produce their eggs and sperm. And so they're actually going after a gene drive approach where they're trying to actually make those heritable mutations and hoping that they'll be able to grow and maintain them. So it's been really great because we have quarterly meetings with this group, and we kind of exchange information and try to assist one another as we all work to solve similar problems.
It's just so much fun. This is great. I really am excited to see this project develop. So thank you so much.
Thank you.